Good morning, everyone.
Good morning.
Here. Let me do a quick test run. Jesse, can you hear us? Calling you out.
Yes, I can hear you guys.
Hey, great. I'm glad you guys can come off mute as well. It's our first run through with the webinar style, so appreciate it. All right, we'll go ahead and get started. Good morning. Welcome, everybody, to the Florida Oyster Recovery Science Community of Practice. This is our fall 2025 webinar. There may be some more folks that trickle in and get settled this morning. So I would ask everyone to please go ahead and share your name in the chat function. It should be available. And then just as a quick icebreaker, tell us the closest bay or estuarine system to where you're tuning in from today. That'll give us a sense of who's here. So for folks I might not have met yet, my name is Katie Conchar. I'm a restoration ecologist with the Nature Conservancy and the coordinator for this FORS COP. I'm based in Tallahassee, Florida, and so my closest home estuary is Apalachee Bay or Apalachicola Bay. I'll remind folks, if you're new, the mission of FORS is to support the recovery and management of Florida's oyster habitat and fisheries with science and through collaboration. So happy to have you all here today. We have some guest speakers with us today for the webinar from the Reefense, a Mosaic oyster habitat for coastal defense project. These folks will present their research on genetics, breeding for disease resistance, and oyster growth, based both in the laboratory and at their field location, Bakers Point in St. Andrew Bay, Florida. There will be some time following the guest presentation for open Q&A. I would ask everyone to please keep yourself muted while the speakers are presenting, but feel free to drop your questions, comments in the chat during the call if you prefer to save your thoughts that way, and we'll be sure to review those as part of the Q&A session. This webinar is being recorded and will be made available to the FORS working group following the call. With that, I'd like to introduce our speakers. The Reefense program was created by the Biological Technologies Office within DARPA, which is the Defense Advanced Research Projects Agency, to rapidly advance nature-based solutions to protect shorelines, specifically at military installations, and also specifically to use corals or oysters as the target foundation species. Rutgers University led a team of 11 institutions working on developing materials to form the base of a reef that would be colonized by oysters to create an ecologically functioning oyster reef with oysters bred to be more resilient to disease risks and grow faster than those that are currently struggling in the system. So today we'll hear specifically about the latter part of that project on breeding more resilient oysters. Even this part of the project, all parts of the project were a large collaboration involving Dr. Dave Bushik as the overall lead for Rutgers University, who's with us today. Ximing Guo, who's also a well-known molluscan geneticist from Rutgers University. Scott Ricard and Mason Williams from Auburn, who produced the oysters and tracked their performance in the field. Jerome Lapierre and Sandra Casas-Liste from Louisiana State University, who conducted lab challenges and assessed disease from the field deployments. So a warm welcome to this team. I'll now turn it over to you, Dave, to go ahead and screen share and talk about this exciting work. Thank you.
Okay. Can you see the screen?
Yes, looks great.
Perfect. All right. Thanks for that nice introduction. And you can see our title slide here that basically describes who all is going to be speaking today to you. We're going to do a little bit of tag teaming here so everyone can talk about the different components they worked on. I'm going to start by giving a little bit more of an introduction to the Reefense program. I'll skip through this because you did a nice job of talking about this pretty well, but DARPA created this program back in 2020 with the goal, as seen here, to develop self-healing hybrid biological and engineered reef mimicking structures to mitigate coastal flooding, erosion, and storm damage. The reason that DARPA as a military organization was interested in this is because just like all of our coastlines, whether it's open to marshes and forests, or whether it's protecting homes and municipalities. The bases are also susceptible to the same um, uh, uh, dangers from climate change and erosional forces and whatnot. And if their coastlines are damaged and it impacts their mission, they can't be mission ready to accomplish what they need to do. So they created this program to sort of accelerate the kind of work that you're all familiar with that's been happening with living shorelines, and um, bring together a large team to address several aspects of that. So here was sort of the Reefense program. It was supposed to be a 5-year program in 3 phases, but after the new administration took over, there were a lot of budget cuts, and our program was susceptible to that budget cut. So we haven't been able to complete the last phase. We only did a little bit of it. But we will tell you all the progress that we did made, which was pretty substantial. So in phase 1, when they created the program, there were 3 technical areas. The first one was substrate design and structure. So they wanted us to develop low carbon materials, low carbon footprint materials, and then design its structure based out of those materials. They want us to apply ecosystem engineering methods to make a functioning ecological habitat. And then they wanted us to apply adaptive biology, which is basically the genetic selection that we're going to talk about today, to improve the resilience of the foundational species, which in this case was oysters. After phase 1 was completed, they sort of restructured this. They kind of got rid of these and put them into more of a overarching design. So phase 2 and 3, phase 3 had these more holistic metrics, in which they were concerned with shoreline protection, durability, and cost. And this is kind of how they related back to these different technical areas here. We have created what we are calling a living shoreline mosaic. I know our project was called a Mosaic oyster habitat, but we don't think it needs to just be oysters. It can be any component that's in there. In this conceptual model, we also designed these things called a Reefense module, which is basically a wave attenuation device that we've modified to improve the attenuation capacity of it and also the habitat characteristics of it. So that as soon as it's installed, it will start attenuating waves. So these are the reef materials here. They're designed to with these holes in it and with these ledges in it to hopefully facilitate growth and attachment of organisms, in this case oysters. The materials are designed to last 15 to 20 years at least. We had one component that was one design that was biodegradable and would last that long, and another one was designed to exist much longer than that. As soon as they're installed, they start to attenuate the wave energy. And then behind that, you start to see sediment deposition, which is something we want to happen. The reef, oh, sorry, over here. Behind that, then we hope that other mosaic habitats will grow, which might be additional shellfish reefs, or it could be SAV, or it could be marsh grasses. But these are what comprise the mosaic of the living shoreline. I don't think any of this is particularly novel to anyone who's worked on living shorelines. And then we want the reefs to be durable over time. So those are the basic components of this program. Here is an aerial image of what we installed. So these are the modules out here forming a breakwater reef, and then these cusps and other structures behind it are designed to help reduce some of the flow behind here, the longshore currents, and establish areas where SAV can develop and form. And then we have these cuffs here that are designed to help protect the development of marsh grasses here. As you can see, it was a comprehensive project with 11 different institutions involved. They're all indicated down here. And you're going to be hearing from folks from 3 of those institutions today. For the genetics component of this, we were specifically asked to target Dermo disease. which I suspect most of you are familiar with. It was discovered in the Gulf of Mexico or Gulf of America, whichever you choose to call it these days, in Louisiana. It was associated with oyster mortalities about the same time that there was a lot of offshore drilling happening down here in the Gulf. The oil companies were blamed for it. Turns out that it was not a result of any of the oil activities there, but there was this parasite that was discovered, which is now known as Perkinsus marinus, that causes Dermo disease. Here's a picture of Perkinsus in a histology section in an oyster. One of the common ways we detect it is to put it into a media called raised fluid thioglycolate media, or RFTM, and it develops into these pre-zoosporangia. So for size reference, these are probably 5 to 10 microns in size here. These ones they're different magnifications. This large one here is probably 80 to 100 microns in size. So when we put these small cells into this media, it enlarges and we can stain it black with iodine and then detect it relatively easily. Shortly after its discovery, it was found along most of the Atlantic coast, as you can see here in the red line. And it persisted in that until the 90s. And in the 90s, following a warming trend, it began to enter the Delaware Bay, move up the Chesapeake Bay, enter the Delaware Bay, and move up the Atlantic coast. It's now, in the last couple of years, been detected up in Canada. So it continues its northward expansion there. Resistance and tolerance, those are two different components of being able to combat the disease, are multigenic traits that make breeding difficult. So despite its discovery here in the late 40s, and despite lots of efforts to breed things until recently, there hasn't been a lot of development in advancing the resistance or the tolerance to this trait. And as a result, it remains a major threat to wild and cultivated eastern oysters. And so from that, I think I can pass this on off to Shming. I believe it's Shming that's going to talk next.
Thanks, Dave. As Dave mentioned that Dermo disease is a multigenic tree or polygenic tree. That means it is probably determined by many small effect genes, not like MSX, the other disease. The other disease have a very strong inheritance. So 5 generations of selection for MSX resistance improved the survival by 800% or more. But for Dermo, we have been selecting for Dermo resistance since 1990. Still, after almost 30 some years, the improvement in field survival still like 20% up to 50% increase in survival in our disease resistant lines. So the farmed oysters, our disease resistant lines, still susceptible to dermal mortality. So when a disease is controlled by many small effect genes, genomic selection is more effective. So genomic selection is different from traditional selection. Traditional selection, you pick the survivors, you breeding for the next generation. In genomic selection, we genotype the oysters, we build a genetic model. We start with a training population. In this training population, we know which oyster is resistant, which oyster is susceptible. We genotype them, with thousands of genetic markers, and then we build a prediction model. In the breeding population, you don't really need for the mortality to occur anymore. You can genotype the oysters in the breeding population based on their genotype and based on the prediction model. You can predict their genomic estimated breeding value. We call them GBVs. And then we select the oysters based on their breeding value. These have several advantages. The first advantage is it uses all the variation in the genome, no matter they are big or small. And another advantage is that you can do the selection without wait for the mortality to occur in the field. So you can speed up the breeding very quickly. So genomic selection is more accurate, more efficient. For genomic selection to work, we need a genetic tool. In this case, we need a SNP array or DNA array. So there's a consortium of Eastern Oyster Breeding Consortium that's consisted of 11 institutions from North Carolina all the way to Maine. So the consortium developed a SNP array. Basically, the SNP array looks like the plate on the bottom. It have 384 squares. Each of this square can genotype our oyster. On each of these square, there are 66,000 genetic markers, probes for them on each of this square. So we can extract DNA from the oyster, hybridize them on the array, and after hybridization extension, we can separate the genotype of each oyster at these 66,000 markers into AA, BB, AB. So that's how the SNP array works. Next. Next, Dave.
I'm trying, it didn't click. There we go. Sorry.
So for dermal resistance, the experimental design is that we start with a population, we challenge them with dermal, and after the challenge with dermal, we separate them into dead oysters that we regard them as susceptible, and we select the survivors we think they are resistant. So we take tissue samples from both populations. Those will be the training population. We genotype all the live and dead oysters from the challenge experiment with this SNP array. And based on the genotype, we built a prediction model. We then genotype another sample from the base population, and we predict their GBVs based on their genotype. Based on their GBVs, we select the oysters with the highest GBVs. We call them genomic selected, or FLGS. And we also selected some oysters with average GBVs. We call them genomic controls, or FLCs. And we also kept a phenotypic selected control, which is just the survivors from the challenge experiment. So we did this over 3 generations. We started in 2022, and the last generation was finished was spawned in 2024. The data we are going to present, we focus on the F2 population. Next. Yeah, those are the challenge experiment conducted by Jerome's lab at Louisiana State University. Jerome, you want to take over?
Yeah, sure. So yeah, so we have for the F2s, we had FLGS, 1,000 oysters, FLGS and FLP. Those were acclimated in the lab, tagged all the way to recruited, and then we injected them with cultured parasite, Perkinsus marinus. They were monitored daily. We recorded the time of death, measured the infection intensity, and all the dead oysters we saved a piece of metal tissue for genotyping. Once mortality reached about 30–40%, depending on the line, some of the survivors were sacrificed and genotyped, and the rest of the survivors were kept to produce the F2 phenotypic cell line, the oyster line. And at the same time, we also had some some of the FLGS and FLP breeding population that were kept in Alabama. And those were tagged. And we also collected a piece of manatee tissue at the Shellfish Lab in Auburn. Those were sent to for genotyping, and based on the training population, breeding values were estimated to produce the FLGS 23 and FLC 23. The genome is selected and control. Next.
Yeah. For data analysis, this is a little bit technical. Basically, we have genotype data, 60,000 genotype from about 1,500 oysters. We did a QC, clean up the genotype data, and we look at the genetic structure in those training population, and we conducted genome-wide association study, or called GWAS. This step basically identify which marker are mostly strongly associated with dermal resistance. And then we break up the markers into 7 data sets, different numbers of data sets. And then we did a cross-validation. For cross-validation, basically we use the 80% of the training population to build the model and to predict the remaining 20% of the training population, which we know whether they're resistant or not. We know whether they survived or not. So that's how cross-validation works. So based on the results, we pick which model to use, and then we use the model to predict GBVs for the target population or the breeding population. After we got the GBVs, because we not only want to improve dermal resistance, we also want to improve the growth of the oyster. So we constructed a selection index. Basically, that's 50% GBVs, 50% of body weight. That compromised the selection index. We select the oysters based on this selection index that is weighted 50% GBVs, 50% body weight. Next. So this is the population structure in the population on the left. The main thing we can notice is there's no difference in distribution between the dark circles, which is a target population, and the red circles, which is the training population, which is good. That means these two populations are identical. We conducted a genome-wide association study trying to identify markers associated with dermal resistance. Very few markers that reach that significance threshold. This confirms that dermal resistance is a polygenic trait. So there's no major loci controlling dermal resistance. Next. So on the left graph, this is in the training population, the prediction accuracy of the 6 models we tested. Basically, as the number of markers increased, the prediction accuracy increased, but it plateaued once we passed like 5,000 markers. But at the end, we picked the GBLOP model by Ramo. This model gave the highest prediction with all the markers. And you can see on the right graph, the predicted values from the different models was highly correlated. That means no matter which model you pick, they give you almost the same GBVs. that give us some confidence that those GBVs are probably real, probably not an artifact. And the estimated heritability for dermal resistance is 0.26, which considered as low or moderate. Next, please. Next is the spawning work that conducted by Auburn. You want to take over, Scott?
Sure, I'll take it from here. Just want to give you a quick overview of how we produce the oysters, produce these lines. So once we get the GBEVs from Dr. Guo, we'll go out to our farm site, pull the oysters from a particular batch, like the genomic selected batch. We bring them in, we look for the tags that correspond to the parents that we need to use. Those oysters are scrubbed of all their fouling organisms. We soak them for about 20 minutes in fresh water. That drives out any shell burrowing critters like mudworms. And then you'll notice that each oyster has an ID number. As we're scrubbing them, sometimes those the tags come off, so we relabel them with a Sharpie just so we know which oyster is which. The oysters are left out overnight, left dry overnight, just covered in some damp burlap. Then the next morning, they're placed into our spawning system. That's the picture to the left there. It's a 96 unit system. Each oyster gets its own container. The system has two reservoirs, a hot reservoir and a cold reservoir. So when the oysters are initially put in there, we start flowing cool water over them. We feed them a single cell algae just to get them comfortable and open up, encouraging them to feed and pump. And then we flush that system with much warmer water, and that rapid change in temperature is what triggers them to spawn. So yeah, if yeah, that's a female there on the left, and that's a male there on the right. We'll further enhance, our chances of getting the oysters to spawn. We'll strip some donor male oysters. We'll suspend the sperm in some seawater. We'll microwave that, and then we microwave it to heat it and kill the sperm, but not denature whatever chemical cues there are that will trigger the spawning. So once we start that warm water cycle in the spawning system, we start pipetting in that denatured sperm mixture, and that usually gets things to start spawning in about 20 minutes. Yeah, there we go. That bottom picture there. So to maximize the diversity in our lines, we take all the females that spawn, we pool all the eggs together, we mix them thoroughly, and then we split them back out into individual beakers. The number of beakers is determined by the number of males that have spawned. So then we fertilize each beaker with a single male. So that ensures that every male has a chance to fertilize every female. And so that provides us the maximum number of crosses. All right, let's jump on forward one more slide. Okay, so then once we've got the fertilized eggs, they're pooled back together and distributed to our larval tanks. And in larval tanks, they're fed a single cell algal diet. The tanks are drained every other day for about 10 days. And during that time, we may do density reductions to keep growth rates high. But for, as opposed to like what we do for aquaculture, with a selection process, when we're decreasing densities, we don't do any selective grading. we want to ensure that we're keeping the diversity of the population. So we may reduce densities, but we don't do any selective grading. Once we begin seeing odd larvae or petavillagers, those are larvae that are ready to set. We'll screen those off on a 200 micron screen. That's those in the upper right there. Those larvae are competent to set. So then they move out to our nursery system, the picture in the bottom there. And again, to ensure diversity, we want to set some larvae from every day that petavillagers are coming off. So we set 300,000 larvae for the first 5 days that we grade petavillagers off. When they go into the system, those silos have a very finely ground up oyster shell on the bottom. And we put the larvae in there. At that point, it's a static system. So water's recirculated in there. Again, we're feeding them the algae diet. And each larvae finds one little grain of that ground up oyster shell to set on and essentially produces a single oyster. After 4 days of setting, then they go to flow through, and then they start growing rapidly, feeding on naturally occurring algae at that point. And again, to ensure that we're not that we're keeping our diversity, we estimate the setting percentage of each silo and count them, and then we reduce densities down to 100,000 per silo. At that point, we can pool them for further rearing. We'll move them out to our farm sites and grow them up till they're roughly about 25 millimeters in size, at which point we can start distributing them to our field sites and provide some for Dr. LePere to do the Dermo testing on. Next slide. So Dr. Guo went over some of this. I'll just reiterate real quickly. In August and September of 2023, we produced the 4 different lines. The FLWC line is a random control line from wild oysters from the Baker Point study site in Florida. We've got the phenotypic line, which are the survivors from the Dermo challenge, and we select the heaviest individuals to increase the chance of faster growth. Then we've got the genomically selected line with the highest GEBV and weight index. And then we've got the genomic control line, which is the average GEBV and weight index oysters. Down on the left, this is the GEBVs of the actual oysters that spawn. So we may have 96 oysters in the system. Usually we're targeting like the top 10% for the FLGS line. So it may not be that many oysters in there. It may be more like 48, 50 oysters. Not all the oysters will spawn. So we collect data on females and males of which oysters spawn. And so that there on the bottom left is the GEBVs of the FLC and the FLGS line to show the difference in those breeding values. All right, we can pass it on to Jerome to kind of go over some of the lab testing of dermo tolerance.
Yeah. So in the spring of 24, when the oysters are about 6 months old, we got oysters from Scott and brought them in the lab and had them in 6 recirculating water system with filtration, 10 to 1 micron and UV filtration and biological filters. And so we had those 4 different groups, genomic selected, genomic control, phenotypic selected, and then the wild controlled that were produced from Florida oysters without any kind of selections. There were 6 replicates. 3 of the replicates were of oysters were injected with a parasite by shell cavity injection, and the other oysters from the other 3 replicates containers were injected with a saline. And again, we monitored the mortality every day, and then we sampled some of those oysters on week 1, week 9, and week 18, and measured the body burden, the number of parasites there were per gram of oyster tissue. And this is the results. As you can see, for the Dermo challenged, the Florida wild had much higher mortality and sooner than the Florida genomically selected. The phenotypically selected and the genomic control had about the same level of mortality. So there was significant difference in all the 3. In the saline injected, we did some, at the end of the experiments, we did some, there was some mortality, but it was not significant. If you looked at the infection levels on those oysters, there was no significant difference. However, there was a trend of the Florida, the progeny of the wild oysters from Florida had tended to have much higher heavy infections of Perkinsus marinus. So that was the results. So there's definitely a trend of heavier infection in the Florida wild, which Mason was going to talk about later, but in the field, you see some drastic differences also. The next slide. Yeah, and I guess that's a summary slide in terms of percentage. The genomic selected had a 60% higher survival than the genomic control. FLP, the phenotypic selected and the genomic control didn't show much difference, but the most striking difference was the genomic selected had a 240% higher survival than the progeny of the wild Florida oysters. So in terms of improvement of the F1, had a 10% improvement, the F2, 60% improvements, and improvement over the wild, the progeny of the wild oysters was 214%. So in conclusion, the genomic selected can significantly improve dermal resistance, and it's more effective than the phenotypic selection. You want to go ahead, Mason, and I guess you can talk about the field testing?
Yeah, so for the field component of this project, we tested the 4 oyster lines across 4 sites, each representing different environmental conditions. First, we had Baker Point within East Bay of the St. Andrew Bay system. This was the intended deployment site of the improved oyster lines in the reef. And then we had the FSU Panama City campus site within North Bay of the St. Andrew Bay system. And then in Grand Bay, Alabama, we had oysters there. That is the Auburn University Research Farm within the Mississippi Sound. And then lastly, Grand Isle, Louisiana at the Voisin, Louisiana Sea Grant Hatchery. At each site, we deployed 5 replicate baskets per line with 120 oysters per rep, so 600 oysters per line at each site. The baskets were enclosed and suspended off bottom to ensure mortality was not caused by predators such as crabs and conchs. And we collected monthly shell height and mortality measurements plus quarterly dermo infection intensity. And yeah, that video shows our FSU site for the baskets suspended off their pier. But we can go to the next after. Looking at mortality, both the genomically selected high breeding value line FLGS, and the average breeding value FLC line had lower mortality compared to the phenotypic and wild control lines at the target reef site at Baker Point. When looking at the FSU site, we can see a spike in mortality in the summer of 2024, which was caused by me installing the lines during high tide. And so during summer low tide, they were exposed to the hot Florida sun and baked a little bit, which explains that steep initial mortality. At both Grand Bay and Grand Isle, we see some separation between the genomic select lines and the phenotypic and wild control lines starting in the summer of 2024. However, a sudden unusual mortality syndrome or SUMS event in the late summer of 2025 caused a rapid mortality event, converging all lines and masking earlier performance differences. And then we can go to growth. Again, the only statistical difference was found at Baker Point, with the wild control line growing significantly slower than all lines. Growth in Alabama and Louisiana were calculated only up until August 2025, right before the mass mortality event wiped out most of the oysters. And though not significant at all sites, the overall trend shows that the selected lines maintain a growth advantage over the wild control line. And then we can go to field dermo infection. Quarterly dermo infection intensity analysis shows that most sites maintained relatively light infections, with the Alabama and Louisiana sites showing the highest overall infections. In general, the sites maintained very low dermo infection levels across the 2 years of tracking, with the exception of the FLWC line in last July's sample. While we see more progressive infections as the oysters get older in Alabama and Louisiana. Across all sites, the wild control line consistently had higher infection intensity compared to the other lines. And we are processing our final samples for these sites, or LSU is processing these, the final samples from our October sampling. And hopefully that will kind of clear things up. And then together, these results reinforce that the selected lines are showing gains in survival, growth, and disease response relative to the wild stock. Go to the next. I believe this is Dr. Guo.
Okay. In order for us to deploy the genomic select lines in Florida for evaluation, we applied a permit for deploy them. To show that the genomic select oysters are not monsters, we analyzed their genetic differentiation from the Florida wild population. So we analyzed those populations, this FLW0, or those are the wild Florida oysters collected from Florida. The FLW1 are the F1 wild, W2 are the F2 wild. The GS1, 2, 3 are the genomic selection generation 1, 2, 3. The GC is genomic control from the third generation. The FLP3 is the generation 3 of phenotypic selected. From each population, we sampled 65 or so oysters. We estimate the effective allele number. You can see the third column. They are very similar. The Florida wild population have the highest allele diversity, but there's a slight decrease in the allele number. The heterozygosity is basically the same. The FST statistics measures is whether these populations are significantly different from the Florida wild. You can see the numbers are very small, not significantly different from zero. That means overall there's not a significant genetic differentiation between the selected and the Florida wild. But when you look at them at a genome level, you can detect some subtle differences. On the right is the PCA plot, which show the different populations. The small red or pink dots in the middle are those, yeah, those are the Florida wild population. And the ovals on the right are the F1 and F2 Florida wild. You can see even, well, the F1, F2 Florida wild are from a different selection. That means even if you just grab some oysters from the Florida wild, you produce them in the hatchery, you create some difference because of genetic drift. And so this is very typical in any hatchery. Anytime you grab oysters from the wild, reproduce them based because of genetic drift, you will create some genetic variation, but those variations random is not significant. All the other circles are the selected oysters. They're not uh, significantly different from each other. They're slightly different from the wild, but overall not significant. On the left bottom are those the estimates of inbreeding level. You can see for the wild populations, the inbreed level is ranging from 1 to 2%. In the selected populations, is typically 1 to 3%. So there's a small difference, but the overall the inbreeding is very small. It's typical of what you would expect in any hatchery produced lines, especially after 3 generations in the hatchery. So we conclude there's no significant difference overall. There are subtle differences, which is consistent with genetic drift. So we think you know, those genomic selected lines won't change the genetic structure in the wild population if they're used for restoration purposes. There have been increased interest in using the disease-resistant lines for restoration elsewhere for oysters. Basically, this data shows our genomic select lines after 3 generations, there are small differences, but those differences are not significant. Next.
Okay, I guess that's back to me. So based on that data that Ming just showed, that we really didn't change the genetic diversity all that much, we were able to get a permit to deploy oysters. And so prior to that, we were using the wild oyster spat, which hadn't been selected, to just test methods to deploy things on the oyster reef. And the thing that worked best for us was to set oysters onto these things called tufts that are oyster catcher tufts that are produced by Sandbar Oyster Company out of North Carolina. And it's basically just cement coated jute that's kind of wadded up like this. So that structure is solid that's being held right there. And you can see all the oyster spat on it. And we took those and we just ran a little bamboo stake through it and looped them on there. And so long as we pack them inside of the holes here, we would come back and see this. And if we only put one of them in there, the wave action usually just kind of tore this up, and we came back and it was all shredded apart and no longer there. But if we put 5 to 10 of them in here so that the wave action wasn't moving them around too much, when we came back a couple months later, you could see the oysters here that had grown and survived quite well. And that proved to be pretty much the best method that we could do with them. We then, at the very end of presenting that data, got permission to deploy the last group of genetically selected larvae. And so we did that by harvesting the eyed larvae. So Scott and his team harvested the eyed larvae. And this is Mason and I with eyed larvae in a bucket and a siphon. And so Mason's just holding the siphon down along the breakwater reef there. We stuck some tiles out to see if we could capture any of that. And here the next day we recovered the tiles, and you can see the eyed larvae had set and are attached onto those tiles. So we think that this is a reasonable method to go ahead and quickly deploy them on there. Either one of these worked pretty well. You could set structures in the laboratory before deployment, but that seems like a more difficult way to do it with a lot of risk of larvae. And I think that pretty much concludes our talk here. We just wanted to acknowledge the Center for Aquaculture Technology for doing a lot of the genotyping for us, and then our funding sources here, DARPA from the DoD, ASMFC, and NOAA and MSS and the Department of Commerce for supporting a lot of the other genetics work. With that, I think we're happy to take any questions if there are any.
Thank you, Dave and all. I'm sad for that little oyster there who's feeling not strong enough. Thank you. That was very, very interesting. Do folks have questions? And I think just to get the juices flowing, perhaps Dr. Guo mentioned this. I was wondering how this team envisions that the oysters selected under this work could be used to enhance restoration success, and if you considered that would be appropriate only for the St. Andrew Bay system where the breeding population was selected, or perhaps the larger geography where the field testing was done.
Yeah, I see there's no problem for use these stocks for restoration in Florida. Whether it should be used for restoration elsewhere, like on the Atlantic coast, that's probably I would not recommend that because there are other stocks more suitable for use for restoration along the Atlantic coast. But certainly, we have yet to see a really significant market increase in field survival. There are some increase in field survival, not as strong yet. We should continue improve their field survival. Once that's established, improved, I think this should be a good choice for restoration in Florida. Not sure about elsewhere. What do you think, Dave?
I think that there's no problem using these lines or any other lines that are either locally selected or however you want to do it. The genetic concerns are a matter that needs to be decided by the local regulatory authority, I think. We did find that maybe Scott can comment on this a little bit better, that Auburn has a Gulf-wide line. And we did test that alongside of this initially, but it didn't perform as well as the local line did, at least in terms of Dermo resistance. And there may be some reasons why that was so.
Yeah, I'll just comment real quick. Yeah, so the Gulf wide line that we used was a line that we had selected for shell shape and fast growth, and for aquaculture for oyster farming. And it was I'm trying to think now. I want to say it was probably a fourth generation animal, so it had been selected down pretty far. So it's I think we've shown a proof of concept here that you can kind of target a local population and quickly get some improvement in dermo resistance very quickly. Down here in the Gulf, we can turn over a generation every year, so we can move pretty rapidly on these selected populations.
There's a question in the chat from Vincent that Scott and Schming can address. Are genomically selected lines also being used for aquaculture?
Yes. Independent of this study, we have been doing genomic selection at Rutgers for dermal resistance for field survival using genomic selection. Those genomic select oysters are being tested. Some of them are being used for aquaculture. And also from this project, I personally believe the genomic selected lines from the DARPA project is quite valuable because they demonstrated strong thermal resistance in the lab, although the survival in the field is more complex. So we brought these oysters to Rutgers. We made a hybrid between the genomic selected Florida lines with our NEH, which have a strong MSS resistance. We're hoping this hybrid will inherit MS resistance from the Rutgers NEH and the thermal resistance from this genomic select lines from DARPA project. So we made that hybrid. We're going to continue evaluate them, monitor them. Hopefully, that will provide a better line for aquaculture people to use for production. Similar efforts may be going on in Auburn. Scott, maybe you have some efforts there also.
Yeah, so we are testing some of these in an aquaculture setting. This year we produced some triploids made from this broodstock line. If you're familiar with triploids, they're relatively sterile animals, so they tend to grow rapidly and have a good meat yield in the summer when your other oysters are spawned out. So we're kind of comparing the diploid line versus a triploid made from the line and see how they perform in an aquaculture setting.
Excellent. Thank you. Any other questions from the group? Well, I thank you all very, very much for taking the time to come to FORS and present this information. We've been looking forward to the results of this genetic work for some time, and I think it has great application for folks not only in Florida but across the Gulf. Stay tuned, members of FORS, for a recording to come out shortly and invitation to our spring 2026 event, maybe a webinar, maybe taking another form, but we'll send out that information shortly. And with that, I wish everyone a happy upcoming holiday week, and thank you again for attending today.
Thank you.
Thank you.
Thank you very
Bye,
much.
everybody.
Very interesting.
Thanks a lot, Katie.